Greetings, my fellow biologists. Welcome to Unit 3. In this unit, we'll be going over cellular energy, or how cells maintain their existence. Make sure your guided note packet is out with the writing utensil, and we are going to jump right in to Lesson 1. And Lesson 1 goes over cell transport and homeostasis. Cell transport means how cells bring in materials into themselves to survive or release those materials that they no longer need. With homeostasis, this is how our cells maintain the conditions that they need to support themselves, just like we learned in Unit 1. In class, we talked about the different organelles that make up a cell. An organelle is a tiny structure within our cells that help them carry out their functions. To get the full list of organelles that we discussed on the first page of your guided notes, we will discuss them throughout the entire unit. But to start off, we are going to talk about the cell membrane and the cell wall. We need to know that all cells are going to have a cell membrane, and these cell membranes protect the cell from all outside things. So in order to get into the cell, you will have to go through the cell membrane, and it's semi-permeable. Semi-permeable means that it allows certain things to come in and certain things to go out. The cell membrane is made up of protein lipids, which are just made up of fats and sugars that allow things to properly go in and out of the cell. The cell wall is the other structure that I'm going to talk about. Not all organisms have a cell wall. Cell walls are specific to plants, fungi, and some bacteria, and that's because they need that extra support. It's going to give to the cell membrane. We're going to discuss it a little bit further later on in the lesson. We can see this in plants. Plants have a cell wall for that support. Now, in order for the cells to get the necessary materials they need to create energy, they have to be able to move things across the cell membrane. One way they are capable of doing this is through passive transport. When you think of passive transport, think of something being easy like a lazy river. In a lazy river, we're flowing with the path of least resistance. We don't really need any energy to float down the lazy river because we are moving from a high to low concentration, essentially, just like passive transport. And that's a very important fact to remember, that passive transport, no energy is required. So if you take a look at the image that you see on your screen, this symbolizes passive transport. We have particles that are at a high concentration, meaning that we have a bunch of particles in one space, and they are going to move to a lower concentration or get more air essentially for themselves and move to a place where there's not a lot of particles. We want to reach equilibrium. And when we mean equilibrium, that means that both sides of the cell membrane are equal. So again, let's highlight our important information. Extra note taking is always encouraged when it comes to your guided note packets. If you need to make extra notes on the side of your guided note packet, absolutely go for it. The more notes you have, the better. This helps you be successful throughout the unit. So our passive transport requires no energy because particles naturally want to move from an area of high concentration to low concentration. There are two types of passive transport that we're going to cover. The first type is diffusion, and like I said previously, we are going to move from a high to low concentration. Now, with diffusion of water, there is a specific name for it. It's called osmosis. I want you to put a star next to that in your notes. Whenever water is involved, it's more than likely going to be osmosis. So the diffusion of water from a high to low concentration is called osmosis. Facilitated diffusion is another type of passive transport. When you think of facilitated diffusion, think of a helper or helping make something easier. Facilitated diffusion needs that extra help for the particles to move across the cell membrane because they're a little bit bigger than our natural flow. So they'll need assistance of a protein that'll be moving them from still a high to low concentration, and no energy is required. It's just a little extra push. Because we are learning about particles moving across the cell membrane, it's important that I take a moment to talk about tonicity. Tonicity has to deal with solutions, and we need to know what those solutions are. So a solution is simply two or more substances that have been mixed evenly within a liquid. When we dissolve things to the point we cannot see them within the liquid, that's our solution. So in this class, our liquid will always be water. When you get to higher courses like chemistry, you'll learn that you can dissolve things in other substances. There are two parts to solutions. We have the solute, and then we have the solvent. The solute is what is being dissolved, and the solvent is what is dissolving. So I'm going to repeat that one more time. The solute is what is being dissolved, and the solvent is what is dissolving. So an example would be sugar is a solvent because it dissolves in water. The solute, which you see in this image, would be the water. We are now back on to tonicity. Tonicity is going to refer to the relative concentration of the solution. That is going to determine how much diffusion can take place or osmosis if we are talking about water. There are some conditions when it comes to tonicity, meaning there's different types of tonicity environments that can occur. So we're going to take a look at the different types. The first type that we have is hypertonic. Hypertonic or hypertonicity is when the solution outside of the cell has a higher particle concentration than the inside of the cell. There's more water on the inside of the cell, but water wants to move outside to dissolve the solvent. This is going to make the cell shrink or shrivel up. I want you to take note of the picture at the bottom. It's a cell plant or plant cell. The square you see around the cell is the cell wall. Plants have cell walls because they need that extra support to maintain their structures, and plants are always bringing in water in and out of their cells, so they need that to maintain their appearance. Here's another example of more water molecules on the inside of our cell and a high concentration of solute particles on the outside of our cell. Remember, through diffusion, things want to go from a high concentration to a lower concentration. We are always going to be talking about it in the sense of water. Now, water wants to move from a higher concentration to a lower concentration. The water wants to move outside of the cell and it will shrink. The next type of tonicity I'm going to cover is hypotonic. Hypotonic or hypertonicity is when the outside of the cell has the lower solution concentration than the inside of the cell. So the water wants to run inside of the cell, causing it to swell up. Here we have another image of hypotonicity. I want us to take a note of that. When the water goes inside of the cell, it can swell too big. If the animal cell can't hold all of the water, it can burst, but this is where the cell wall comes into play for plants. Plant cells are way more protected than animal cells are because of those cell walls. Their cell wall will kind of squish that expanding cell so it can keep its shape and stop it from bursting. The last type of tonicity I'm going to talk about today is isotonic. Isotonic or isotonicity means the same. So basically the water is moving in and out of the cell, maintaining the perfect balance or equilibrium, and that's what we're looking for in the perfect world, is an isotonic state or environment. Here's another picture. You see specifically for isotonic that our arrows are pointing in and out because we have that balance. There's an equal number. On this slide, you'll see that we have three types of tonicity that can exist for a cell: hypertonic, hypotonic, and isotonic, and the three different ways they affect the cell. Hypertonic causes the water to leave the cell and shrivel up. The cell is trying to balance out the solute on the outside. Isotonic is where they're equally coming in and out of the cell. They're the same. And then we have hypotonic, the solute on the water, or the water comes into the cell, making it swell up. Let's do some practice with what we've learned so far. So we want to go through and see which tonicity affects an animal cell and a plant cell. For hypertonicity, will cause the cell to shrink for both plants and animals because there's more water on the inside of the cell. The water is going to want to move outside. Please make a note that the reason why it's shrinking in your notes is because the H2O wants to move out. And then we have isotonic, and that means stays the same. The water moves in and out of the cell, keeping the perfect equilibrium or balance of the cell. And then lastly, we have hypotonic. And when we have hypotonic, both for plants and animal cells, it's going to swell. The water is going to move in and out of the cell. The main thing that we need to know is that animal cells are less protected against tonicity changes because we don't have a cell wall. Plant cells have that extra layer of protection. We have some more practice problems with tonicity. On this page of your guided note packet, you will need to identify whether each of these squares is depicting an isotonic, hypertonic, and hypotonic environment. And what you'll have to do is draw arrows to show the direction the water is moving and whether the cell will shrink, stay the same, or swell. We'll work through the first one together, and we have some helpful keys that tell us what water looks like and what the solute looks like. And we're going to make additional notes in our guided note packet to help us complete this packet. Step number 1 is to count up the inside number of solute particles. Step number 2, count the outside number of solute particles. Step number 3, draw your arrows pointing from a high to low concentration. Step number 4, identify if it is hypotonic, hypertonic, or isotonic. If we take a look at the first square model, we're going to count up the inside number of solutes of the cell, and that's 4. If we count the outside solute particles, we will see that we have 2. I also labeled that that would be our low concentration of solutes. We always want to think about these problems in terms of what is going to happen with the water. And remember, diffusion is when particles move from a high to low concentration. Osmosis is the movement of water from a high to low concentration. The water particles are greater on the outside, so the water wants to start to move in. So we're going to draw our arrows in. Pointing is going to represent that our water is moving in. If the water moves into the cell, it's going to swell, and this is hypotonic. Make sure you complete the other 2 models before the next class. We finished talking about passive transport across the cell membrane, which does not require energy. Now I'm going to cover active transport. Active transport is when particles go from low to high concentration. Energy is required for this because the particles are going against the natural flow. There are 2 types of active transport. The first one I'm going to talk about is the use of protein pumps. Protein pumps are going to be embedded into the cell membrane. Take a look at the image that's on your screen. We have this protein pump, and its job is to push or pump ions across the cell membrane. We are looking at the pump taking the hydrogen from the low to high concentration where all the other hydrogens are, and they need to use energy, which is the ATP you see in the image. ATP is what energy is in the cell, and we are going to learn more about that later on. The second type of active transport is bulk transport. Think about a bunch of material being brought in and out together in one large mass. Bulk transport is using membrane-bound vesicles to help transport materials inside and out of the cell. The vesicle will fuse with the cell membrane and the material in and will take the material in and out of the cell. There are two types of bulk transport. The first is exocytosis. Think about exiting when you hear exocytosis. We are moving material outside of the cell. Endocytosis is the moving of things inside or into the cell. The cell is going to pinch things together to bring them in, and we have some images on the right-hand side of your screen that will help you understand endo and exocytosis a little bit better. We have another practice that you'll need to complete before next class. Use the word bank provided to fill in the blank boxes. Think about the images that you see here and which ones require energy and which ones would not. Of course, you can use your guided note packet as assistance. Our end result of all of this is homeostasis. The main reason why cells undergo passive and active transport is to help them maintain balance within their cell. Our cells are moving things constantly across their cell membranes so they can maintain homeostasis. The tissues and organs that these cells make up work at their optimal level. That's all homeostasis is. The steady internal physical and chemical conditions maintained by living systems, just like we learned in Unit 1. If it is cold outside, we maintain homeostasis or respond to the environment, we'll start to shiver. If it's hot outside, we'll sweat to cool off. Our cells are regulating water levels, maintaining our calcium levels, making sure that our blood pressure is good. All of these things help us maintain homeostasis. We are at our final practice. We are going to draw a line from a type of transport, and then we are going to say whether or not it requires energy or doesn't need energy. So let's start off with question number 1. We have exocytosis. This needs energy. So I drew a line from exocytosis to needs energy. I want you to keep going and to complete all the practices before next class. I want you to remember that practicing will help you remember the information for your quizzes as well as your tests. If you need any assistance, please stop by Lab 6 for help. Have an amazing day.